Good morning. As I introduced, my name is Adrian Lennon. I'd like to talk some work that we've been carrying out over the last couple of years. It derived from a sabbatical I took 2 years ago, and in collaboration with Dr. Farrell, who's a plant physiologist in DLS, and also with Prof. Uma. Vanessa Lewis joined us as a intern for a summer and then carried on as a project student and is now running with the project as a graduate student for MPhil work. So what we're discussing really is responses to highlight in Theobroma cacao. Now, as everyone's aware here, it is a shade-tolerant tropical tree, and it has a reported light saturation point of approximately 400 micromoles per meter per second of photons. What does that mean? It means the amount of light it can actually use is very low. Now, to put it in context, if you went outside at lunchtime on campus in Trinidad, the light the trees will be seeing there is 1500 micromoles per meter per second. So the amount of light the tree can actually use is very limited. It's also been shown that if you expose cacao leaves to higher radiance levels, then you can actually get a reduction in the photosynthetic rate. So this is the problem we have here. It's a shade-tolerant tree, and it performs poorly in high light. But recently, there's been a trend in some places to move from an agroforestry system to an orchard system. Now, I'm not proposing either is better than the other. Agroforestry has its advantages and orchard systems has its advantages. But the main thing I want to point out is that if you're going to go with an orchard system, you're trying to grow a shade-tolerant species under high light, which will have implications. Now, there is still debate as to whether growing cocoa under full light or shade, which one leads to higher yields. The literature is kind of grey around this area. In some studies, higher light is associated with higher yields, whereas in other experiments, projects, it's shown that yields decline under higher light conditions. Also, production modelling has shown that heavy shade can reduce bean yield by up to 60%, whereas light shading did not affect the yield. With all this in mind, the development of cacao accessions that can withstand high light levels and also produce increased yields is therefore of interest, and this is where this project came from. Now, to remind you of the horrors of your undergraduate biochemistry, I put in the Z scheme. This is what we're talking about when we're talking about photosynthesis. We have the two photosystems, photosystem 2 and photosystem 1, linked in series through another protein complex, cytochrome B6F complex. And essentially what happens is we split water to produce electrons, and the two photosystems raise those electrons to a higher energy level, and the electrons are passed between the systems, moving upwards to higher energy level status where eventually they're used to reduce NADP to NADPH. Whilst this is going on, we generate a proton motive force through proton pumping at B6F, and also the splitting of water, and also the uptake of protons, the reduction of NADP. And the NADPH and the ATP, the products of this Z scheme, are used in the Calvin-Benson cycle to fix CO2 into sugars. Now, we always think of light as being beneficial to plants, but it's also majorly problematic to plants, particularly when you're under high light conditions. So under high light conditions, plants must do two things. They have to efficiently utilize the solar energy, i.e. they have to absorb it by their photosystems, pass those electrons down to form the reductant NADPH and produce the ATP. But while doing so, they generate a very energized system, and they therefore have to avoid photo damage to their cells. Lots of light, high light can lead to photo damage. Now, energy that they can't use for electron transport, this excess energy, they have to dissipate, get rid of it to avoid damage. And it can be done by two ways, either what we call photochemical or non-photochemical, which we call NPQ mechanisms. Now, photochemical quenching, as its name suggests, is passing electrons through the photosynthetic apparatus. So by maintaining electron flow through the Z scheme, as I talked about previously, we can remove the energy from this over-energized system and allow the plant to quench the energy. There's other routes you can use in photochemical quenching. You can have cyclic electron flow. You can use things like the Maillard reaction and also photorespiration, also the chlororespiratory pathways. But using these additional routes beyond the Z scheme can result in the generation of reactive oxygen species, which I'll talk about in a moment. Non-photochemical quenching is dissipating energy, getting rid of it, but not involving this electron transfer. And there's a number of mechanisms that can do it: the state transitions, photoinhibition, protonation of the photosystems. But one of the major ones is using carotenoids, these pigments, to throw the energy back through what we call the xanthophyll cycle. And we'll be talking about carotenoids as we go through this talk. Now, high light can lead to photoinhibition, and it can lead to the generation of reactive oxygen species. So when all these protective mechanisms are overwhelmed, you generate ROS. This means electrons, free electrons, are passed to oxygen to form reactive oxygen species. And as their name suggests, they're very reactive. And what can they do? They can go on to damage cellular components such as DNA, RNA, proteins, and lipids, the membranes. Now, what happens when you generate ROS within a cell? Well, one, you can have membrane damage, which can lead to electrolyte leakage. Now, electrolyte leakage occurs under these conditions, but most of it we now know is not actually membrane damage. It's actually ROS signaling, which leads to the opening of channels, potassium channels in the plasma membrane, which leads to electrolyte leakage and signals to the plant cell that it needs to adapt and repair. It's what we call metabolic adjustment. The plant changes its metabolism to adapt to the stress and repair the damage caused by the stress. This, of course, will have an energetic cost. And if ROS generation becomes too high, particularly under extreme stress, you can trigger programmed cell death and the cell will commit suicide. So ROS can be a problem. It's going to be generated in energized systems. So plants also have systems to overcome the ROS that's produced. And 3 enzymes we've been looking at in our studies, and these enzymes are part of this ROS protection system. The ones we looked at were superoxide dismutase, SOD, catalase, CAT, and ascorbate peroxidase. Now, SOD is the first line of defense against superoxide, and it takes this very reactive species and converts it to hydrogen peroxide. And then catalase and APX function downstream of SOD and convert the hydrogen peroxide back to nice, safe oxygen. So we've been studying these effects on cocoa, this highlight, and the methodology we used is mainly chlorophyll fluorescence. Now, I'm not going to go into the details of chlorophyll fluorescence and the way we measure and the technique behind it. I don't have time. If anyone wants to talk about it, please see me afterwards. But we can use chlorophyll fluorescence to measure a number of things. One of the things we can get at is what we call FVFM, and this is a measure of plant stress. And when we do chlorophyll fluorescence and follow this variable fluorescence over the maximum fluorescence, if we get a value of 0.83, the plant is considered generally unstressed, and anything below that indicates a stressed plant, and it also indicates potential photoinhibition. We also use chlorophyll fluorescence to determine light saturation point, i.e. how much light a plant can actually use. We use it to measure what we call quantum yield, which is an estimate of the efficiency by which light absorbed by the photosystem can be passed to the primary electron acceptor in photosystem 2, namely QA. And we also get a measure of the maximum electron transport rate through that Z scheme I talked about earlier. The 2 other components we measured are QL, which is a measure of photochemical quenching, and we also measured NPQ, that non-photochemical quenching. The other things we measured in these studies is the enzyme activity of APX, CAT, and SOD. We also looked at pigment content by measuring the amount of chlorophyll A and chlorophyll B, and also the total carotenoids. We measured lipid peroxidation as a measure of membrane damage by reactive oxygen species, and we also looked at electrolyte leakage. So the first study we did was published earlier in this year in Scientia, and in this we looked at how different leaves on the same plant respond to light. So we looked at the photochemical responses in the shade leaves compared to those in the sun leaves. Now, to do this experiment, we used WAA plants. They're about 25 years old, and they were growing in full sun on our campus at UE. All experimentation through all the studies I'm talking about was done on fully expanded leaves, and they're all at the interflush 2 developmental stage. So the first thing we looked at was photoinhibition, looking at this FDFM value. Now, first of all, I want to indicate the light levels we're seeing. The shade leaves are shown here in the dark circles, the sun are in these light circles. Now, the shade leaves at 8 AM are looking at 43 micromoles per meter per second of light, around about the same at lunchtime, and dropping off to 8 in the evening. So very low light levels. If you look at what the sun leaves are experiencing, in the morning 182, at lunchtime they're about 1500 micromoles per meter per second, and in the evening they drop down. Remember, the saturation point for cocoa is generally considered 400 micromoles or below. So we're seeing very, very high light levels. And if you recall, I said a value of 0.83 and above suggests the plant is not stressed. And if we look at the shade leaves, they are remaining happily through the day, no significant differences above 0.83. These are nice, happy leaves. They're showing no photoinhibition. But if we look at what's happening to the sun leaves, even at the start of the day, we're seeing the values below, well below 0.83, which become lower and lower through the day. This suggests two things. One, it suggests that the plant, these sun leaves, are permanently displaying photoinhibition. Now, there is some recovery at night because we return from this lower level, 4 p.m. back to our higher level in the morning, but it's still permanently photoinhibited. So this is just two things. The recovery is probably short-term acute photoinhibition of photosystem 2, and there may be some chronic long-term photoinhibition occurring as well through maybe photosystem 1 problems. We haven't actually measured that, so I can't say for sure. But the sun leaves are photoinhibited, the shade leaves are happy and sitting where they are, they're quite happy in their life. We looked at light saturation and the electron transport rate, and what we can see is the light saturation point for the sun leaves. They can cope and use much more light, and if you look here, we're about 325 micromoles per meter per second, in the same ballpark as that reported 400 micromoles, whereas the shade leaves are incapable of using much light. They're not very well adapted to use light. If we look at the electron transport rate, the flow of electrons through the photosynthetic apparatus, again, we see much higher levels in the sun leaves than in the shade leaves. Now, what does this tell us? Even though we have photoinhibition in these sun leaves, they appear to be doing much more photosynthesis. And we also confirmed this electron transport rate by using infrared gas analysis, and we also had higher rates of carbon fixation in the sun leaves versus the shade leaves, even though we were photoinhibited in the sun leaves. We looked at QL, photochemical quenching, and NPQ in the sun and shade leaves, and again we see the sun leaves are very good compared to the shade leaves at photochemically quenching. They can dissipate this excess energy by flowing electrons through the photosynthetic apparatus. But if we look at NPQ, at first glance, you'd probably say that the sun leaves, shown here in red, are much better at NPQ than the shade leaves. Well, they probably are a little bit better, but because of the difference in FDFM, it's problematic to directly compare these two values. But what we can say, though, is both the sun leaves and the shade leaves are saturating their ability to NPQ at quite low light levels, here at about 100 micromoles per meter per second. So the sun leaves are actually very poor, it seems, at carrying out NPQ, dissipating this excess energy. Looking at pigment content, we looked at chlorophyll A, B, and total carotenoids and looked at the ratios. The sun leaves and the shade leaves are displaying typical pigment characteristics for sun and shade leaves. So we do have plasticity in the pigments. The plant is able to change such things as its antenna structure, its light harvesting complexes, and its carotenoid ratios when the leaf is exposed to full sun. There is that plasticity. So if we expose a cacao leaf to high light, it will respond in changing its pigment content, which is a good thing if we want to grow it under high light. When we looked at the enzymes, SOD showed no significant differences between sun and shade leaves, whereas APX happily increased, so the plant is able to respond to higher light by increasing APX, and there was also a small but significant increase in catalase, possibly due to an increase in photorespiration. One thing we should note here is that the electrolyte leakage was significantly greater in sun leaves than shade leaves. So even though we have more photosynthesis going on, we are producing more ROS and we have issues with electrolyte leakage. So what can we conclude from this study? Well, first of all, sun leaves have an increased capacity for photosynthesis over shade leaves. However, they are subjected to photoinhibition. The sun and shade leaves display typical pigment plasticity, which is a good thing, but when we compare it to other species, the chlorophyll carotenoid ratio was low for both types of leaves. Plasticity for the ROS scavenging enzymes APX and CAT, but there was no plasticity in the levels of SOD between sun and shade leaves. This increase in electrolyte leakage is a problem, alongside the photoinhibition, which suggests that even though we get higher photosynthetic rates in the sun leaves, the WAA leaf accession in this case is not well adapted to high light conditions. And with this electrolyte leakage, we're expecting that the plants are undergoing metabolic adjustment. And so this metabolic adjustment I mentioned earlier does have an energetic cost, respiration, and it may reduce any of the gains seen in photosynthetic rate. So further studies are required to examine respiration rates, photo and photorespiratory rates. So following this, we carried on and tried out, so there was plasticity within the plant, but what about genetic variation across different cacao accessions? And so this paper was just published last month. It came out in Helion. It's open access, so if anyone wants to get hold of it, feel free. We used 7-year-old accessions that were selected for the study, 3 replicates. They were in the shade house under moderate high light. And if we look at the light values they're seeing, in the morning, about 400 micromoles, at lunchtime, 700, and in the evening, back to about 400. So we're under some level of shade, but it is high light for the plant. This is all above the standard 400 micromole light saturation point. The accessions we used are shown here. We took from a number of genetic groups, a number of accessions. Now, why did we pick these particular accessions? There was no clever reasoning behind it. This were plants growing in the shade house. They've been growing for a number of years under these conditions, and I piggybacked on another project that was running using these plants. There was no rhyme or reason in the selection of the accessions, but we've got a nice distribution of different plants. Now, I'm not going to have time to show the data. It's all in the paper, but what I can say, there was significant variation between all of these accessions for all parameters observed. So FDFM, the light saturation point, the efficiency of transfer of energy from photosystem 2 to QA, the electron transport rate, the pigment content, and the ratios were all different. The enzyme activities, there was a lot of variation there. And again, with lipid peroxidation damage and also electrolyte leakage. So we had variation between the accessions of all these parameters. So there is genetic variation there, which is a good thing if we want to start breeding for plants that can resist high light. When we looked at the correlations, it's quite interesting. It's the correlation table. Shown in red are those that are significant. First of all, we see a nice negative correlation, significant correlation between electrolyte leakage and the chlorophyll to carotenoid ratio. It's a negative correlation. So what does this say? It suggests the more carotenoid content we have, the less electrolyte leakage we have. So that's suggesting the more we can dissipate energy through carotenoids going through the xanthophyll cycle, the less electrolyte leakage we have, which is a good thing. We had correlations between the saturation point and the maximum electron transport rate, and also between quantum yield. So these 3 values here. This suggests that if you have a higher light saturation point, you have a better quantum yield, a more efficient transfer of electrons, and a higher electron transport rate. So measuring the saturation point gives us an indication of the ability to pass electrons through the photosynthetic apparatus. When we look at the enzymes, there was a negative correlation between APX and SOD. There was a positive correlation between APX and catalase. And when we looked at the electrolyte leakage, there was a very strong negative correlation between SOD and electrolyte leakage. So the more SOD you have, the less electrolyte leakage you have. Whereas in the case of APX, there was a strong positive correlation between APX and electrolyte leakage, suggesting the more electrolyte leakage you have, the less APX you have. sorry, the more APX you have. More APX, more electrolyte leakage. So what can we conclude from all of this? So if we're going to try and grow cocoa under an orchard system, then we need accessions that demonstrate high light saturation points, which means they should also have high quantum yield and high electron transport rate. Alongside that, we want them to have limited photo inhibition. So FVFM values closer to 0.83 through the day as we can get. We also note, which I didn't show, that all the accessions we measured, as we've shown here, had AB ratios, chlorophyll AB ratios, consistent with sun leaves, suggesting that just like the WAA, there is the ability to have plasticity to respond to high light. So if I take these accessions and put them under high light, the chlorophyll content, i.e. the light harvesting complex, the antenna structure, will change to be more typically a high light sun leaf. There's also genetic variation in the chlorophyll to carotenoid ratio, and therefore an ideal accession growing at high light intensities would have a high total chlorophyll and a high total carotenoid content. And for that carotenoid content, as it increases, reduces electrolyte leakage. There was significant variation in the enzyme activities between accessions. However, all the values we got in cocoa were low compared to other shade-tolerant species. A good example of that is coffee. So coffee is much better in terms of these enzymes to adapt to high light than cocoa. Cocoa is particularly poor at it, it seems. It's not very good. But there is genetic variation, so there's hope to breed for those with higher, particularly levels of SOD. The strong positive correlation between ascorbic peroxidase and catalase suggests a coordinated response to ROS. Now, APX generally responds to, in the chloroplast, to the QA reduction state, and catalase may be indicating an increase in the photorespiratory flux through the peroxisome, but these two seem to go hand in hand. So higher light, more ROS, more reduction in the system, more carbon flow, both of these increase their activity. There was no correlation between SOD and CAT, and a significant moderate negative correlation between SOD and APX. This suggests that SOD, and cacao at least, is regulated independently of the other two enzymes. So APX and catalase appear to go hand in hand. SOD is regulated in a different manner and is doing its own thing. Now, the significant and very, very strong negative correlation between SOD and electrolyte leakage suggests that those accessions with higher SOD activity may be better suited to high light conditions. We have high light, we're producing ROS. If you've got a lot of SOD, you can clean that ROS away very easily and limit electrolyte leakage, which therefore that limits your ability, your need to do metabolic adjustment and hopefully lead to higher productivity. Now, the APX activity demonstrated a strong positive correlation with electrolyte leakage. And this suggests that in this case, the leaves were responding to the stress induced by high light and increasing APX activity, but it's the levels of SOD that limit the response and the consequent electrolyte leakage. So yes, you're making more APX, but it's the SOD's the problem, too little SOD, and your electrolyte leakage is not going to change. Now, overall, what we can say is if we're looking at a breeding program for those that can look for high light tolerance, you want to look for cocoa accessions with high SOD activity, high carotenoid levels, both of these in relation to that electrolyte leakage, and also high light saturation, which goes hand in hand with high electron flux through the photosynthetic apparatus. Or if you have these, you may well be better adapted for high light conditions. And so what we're suggesting from this study is that using SOD activity, carotenoid content, and the light saturation point may be potentially good markers for the use in screening cocoa accessions for high light tolerance. And that's really our take-home message there. Lastly, I'd like to make a couple of acknowledgements from people who've helped out. Vijay Randan in Life Sciences, the biochemistry technician, and he's been very helpful technical assistance, particularly with grinding up lots and lots of tissue. He's always there for us. And of course, the staff at CRC have been very helpful, particularly the staff of the greenhouses who've looked after plants and made sure they have stayed alive, particularly now when we're all off campus. So thank you to these guys. That's all I have to say. If anyone has any questions, I'm more than happy to take them now. Thank you very much.